Hello, welcome to 13 Universal Learning Beyond Borders. Okay, and this is A-level CIE Biology with Shedro. We are diving into now Animal Transport Series 2. We have had Series 1 and this is Series 2. Please visit our YouTube channels. Make sure you subscribe, like, share, and comment. Now get your writing pad and your pen. Let's dive into the questions. Let's go. The first question says the graph shows the oxygen dissociation curves of hemoglobin for 8 mammals. Wow, 8 mammals. We can see them key mammals in the decreasing order of size. See decreasing order of size. We have elephant, horse, man, sheep, fox, cat. You can see that. All right. So which conclusion can be drawn from the graph? So looking at the graph, which conclusion can we draw? So try it. Try it. Okay. So look at it. If you see this is decreasing order. There's something about their size. Okay. See, of course, this dissociation curve, oxygen dissociation curve, is about the percentage saturation of hemoglobin with oxygen against partial pressure oxygen. So partial pressure oxygen, this is the independent variable. This determines this. Okay, you can see that generally for all of them, as this increases, this also increases. As partial pressure of oxygen increases, percentage saturation of hemoglobin also increases, right, for each of them. Now, I want you to note something. Okay, the one that has, of course, the highest affinity for hemoglobin, we have the highest partial pressure of that it will have the highest percentage saturation of hemoglobin. I mean to say at lower pressure. Okay. Okay. Look, if you pick out, okay, let's pick this 6. Check the 6. See the percentage saturation of 8, which is mouse, is below 50. If you go on, the next one, which is rat, is okay, almost 50. If you go on, see, and of course, you see, the lower the percentage saturation, the less affinity they have. All right, let's go to the question. All right, so if you go to number one, so the activity of mammal is directly proportional to the ability of each hemoglobin to release oxygen. This might make sense, but it's just that we're looking at the conclusion that can be drawn from the graph. Okay, so this doesn't tell us anything about the activities. Is that okay? It doesn't give us ideas about the activity anywhere. All right, that's why we're jumping this. It's going to be the hemoglobin of larger mammals will release oxygen at a higher partial pressure than that of a smaller mammal. If we check the larger mammals, okay, let's check this. Okay, now at a higher partial pressure, let's just check 6 or 8. Okay, pick 6. You trace it, you see that the lower ones, okay, 8 and 7 has the percentage saturation of hemoglobin lower than 50, and that means that they will release more than, okay, so at this pressure it's possible that will release oxygen. But look at, if you get here, the largest animal here, elephant, number one, the percentage saturation is almost 100, so it doesn't release it. Okay, so larger mammals have higher percentage, have higher affinity for hemoglobin. Okay, because they have higher percentage saturation of hemoglobin at low, okay, or higher, okay, of course, pressure. So B contradicts it. All right, says the hemoglobin of larger mammals will, no, they will not release it, because you see, the lower the percentage saturation of hemoglobin, the more readily they release it. Okay, so let's go to the next question. You can see this, the next option. So the hemoglobin of smaller mammals will release oxygen at, now this is a type of error. This is at a lower, at a higher partial pressure of oxygen than that of larger mammals, at higher. Okay, this is the same thing as they just swap it. Okay, it's a type of error. So of course the hemoglobin of smaller mammals will release oxygen at a higher percentage saturation of oxygen, because if you look at here, maybe at 6, remember this is 8 is more likely to release oxygen here than 1 is almost saturated. Okay, so you see that the smaller animals, okay, they combine their percentage saturation of hemoglobin is less, so they have less affinity for hemoglobin, for oxygen. Their hemoglobin has less affinity. Okay, so you see that this at a higher partial pressure, this or this. Okay, that's about 50. But if you check one, which is the last one, is about 100. Okay, I think B is the answer. Then this is the size of mama is directly proportional to the ability of hemoglobin to release oxygen. Well, this did make sense. The size of the mama, okay, is directly proportional to the ability, but that is not what we see here, right? It's telling us that one release oxygen fastest than 8, but you see one has more affinity than 8. That is, number one is elephant, has more affinity for oxygen than 8, which is a mouse. Why? Because if you check the partial pressure 6, take it up, you see that it has highest percentage saturation of hemoglobin with oxygen than 8. One has higher percentage saturation of hemoglobin than 8, so that means it has higher affinity. Remember, the principle is that the one that has higher percentage saturation of oxygen, all right, at a particular pressure, most likely going to have more affinity, all right, so it will release it less. So C is the answer. Let's run. The table describes the wall of three blood vessels. What is, oh, the answer has appeared already. Okay, yeah, so now they say the answer is A here. Good. Why is it A? Okay, A says artery, thick layer of elastic fibers. Yeah, artery has thick wall because it carries blood at high pressure, so it doesn't burst. So the only one that shows artery here is A. That's one as artery is A. Okay. Capillary, no elastic fiber or smooth muscle, has thin wall too. Okay. Yes, so that goes for capillary. And of course, if you check this one, thin layer of smooth muscle with, so thin wall is vein. So yeah, A is the answer. Okay, try this one. Which structures are found in both arteries and capillary? Okay, I want to show you something. Now, if you check artery, okay, I want to show you something. Now, if you look at, let's draw artery first. Okay, let me just sketch it. Sorry. Let me clean it. I try to draw it. I'm gonna try to use the drawing tool. Sorry. Now, artery has thick muscular wall. Okay, because it carries blood pressure at high pressure. Okay, yeah, trying to do that. All right, yeah, thick muscular wall. All right, you see that they have 3 layers. Okay, each of them has 3 layers. Artery and vein. Okay, let me draw vein. Try to draw vein. Oh, all right, vein has thin wall because it carries blood at lower pressure. This vein must have thin wall. It must have it. All right. Sorry for the diagram, but I'm just trying to paint a concept here. All right. Yeah. Okay. Oh, this is supposed to be thick. Now vein has thin wall. All right. Sorry. Okay. Watch this, guys. Yo. This is just one cell thick. Just one cell thick. So figure out which one is that. That is one cell thick. This is just a cell. So this is just nucleus. Okay. Sorry, nucleus just saying the stain and all of that. Okay, look at this, guys. This is capillary. It's one cell thick because the function is the exchange, right? It allows exchange of material between blood inside the lumen here and tissues around it. Okay, so exchange of materials is one cell thick, and these cells are called endothelium cells. Okay, now this is capillary. Sorry, this is vein. Vein is thin wall compared to the artery, thick wall. It has large lumen compared to artery, but both of them have something in common. They have 3 layers. This is outer layer, tunica intima, tunica externa, okay, and the middle layer, the tunica media, and this is the inner layer, endothelium. All right, same thing here. This is tunica externa, tunica media, the middle one, and endothelium, okay, which is tunica intima. So you see the outer layers have collagen and elastic fibers, okay, and both of them help to prevent rupture, especially in artery, busting of artery because of high pressure. Though elastic fiber also helps to, maintain pressure. It recoils and stretches to maintain pressure. All right, both of them have the middle layer, which is collagen, elastic fiber, and smooth muscle. Smooth muscle helps to regulate the diameter, okay, and then control volume of blood flow. And they also have this inner part, which is endothelium. The endothelium helps a lot, right, to smooth out, okay, friction, okay, pressure. Now you see endothelium is here, endothelium is here. This is artery. The inner layer is endothelium. This is vein. The inner is endothelium. See the inner layer endothelium. This is capillary. It's just one cell thick. Okay, that's endothelium. So the question says which structures are found in both arteries and capillary? Endothelium found here, endothelium found here. So what is the answer? Okay, see, let's go. Which function can be carried out by a mature red blood cell? Yeah, so let's talk about that. Mature red blood cells usually they don't have nucleus. The nucleus degenerates. Okay, now that means forget about cell division because cell division is controlled by nuclear division. So they don't have nucleus to create space for hemoglobin. All right, they are not phagocyte, they are not white blood cells, so they don't play phagocytosis. Now because they don't have nucleus, they can't carry out protein synthesis because protein synthesis will require transcription and translation. Transcription will require DNA and they lack nucleus. So B, C, D can be the answer. A, active transport. Here I may have mitochondria for active transport, okay? Yes, so that is the answer, right? Number 5, what is the function of porcine tissue in the mammalian heart? Please, there's only one function for it. A says to conduct a wave of electrical excitation over the atria. No, it doesn't convert over the atria. To conduct a wave of electrical excitation over the ventricle, that's what it does. Okay, it carries it over the ventricles. It's a conducting fiber. Okay, C says to reduce the spontaneous contraction rate of the heart. Nope. It says to separate oxygenated blood from deoxygenated blood. This is done by septum. So now anytime you see porcine tissue, know that it conducts this is a wave of electrical excitation over the ventricles. That is the job of it. Okay, so which type of tissue is present in the wall of all blood vessels? See, I want to answer the question. I drew it last question. Which one? Is it elastic? No, elastic fiber is not found in it's found in it's found in arteries and the vein. Yeah, endothelium is found in everything. Smooth muscle is found in artery and vein. Okay, fibrous, of course, not really. Okay, yeah. So what is you find in all of them is endothelium. All right. The mammalian circulatory system is described as a closed double circulation. The diagram shows the circulatory system in a different organism. The arrows show the direction of blood flow in the vessel. How is the circulatory system in this organism? Look at this organism. Now, this is no longer in the syllabus, but let's see. This is the heart. There's atrium and ventricle. Can you see that? Look, these guys have blood vessels. So because the blood is inside blood, this heart, this particular system has blood vessels, and that allows the movement of blood. Blood is always contained inside these vessels. Always contained inside. Because it's always contained, it is said to be closed. So the blood doesn't back the tissue. So let's check an option that is close. Yeah, A or B. So we eliminate C and D. Okay. So they said now closed double means that it goes through the heart twice, but it goes through the heart once in one circulation. So it can be double. So it must be single. Okay, because it goes past through the heart once. Alright, closed single. What is the state of the valves in the mammalian heart when the pressure in the ventricle reaches its mammalian maximum? I drew a sketch heart in series 1. Should I do it now? Okay, should I okay, allow me to sketch something. All right, just be patient with me. Oh, just okay, or sketch the heart for me. All right, sketch it for me. Just want to show you something. I feel I should show you something, so stay with me, please. I'm just sketching, right? Not real diagram. So I'm drawing. This is the left part of the heart. Okay, yeah, let me draw the right part. The left part and the right part separated by septum. Okay, okay, just all right. So of course, this vessel here, this one is aorta. Okay, and then of course this one is pulmonary artery. They all carry blood away. Now let's get this sorted out. Come with me. Let's look at this. So this is the bicuspid valve that I'm drawing. Okay, separate the left atrium from the left ventricle. All right. Now watch this and prevent blood backflow. Oh, this is okay. Tricuspid valve. Tricuspid is found on the right side. Prevent backflow of blood from the right ventricle to the left. Sorry, from the right ventricle to the right atrium. So this is seminal valve. Okay, it's a pulmonary. So it is called pulmonary. But this is seminal valve inside it. Okay, prevent the backflow of blood from aorta to the left ventricle. All right. So you see, blood returns here. Deoxygenated blood. This is the right part of the blood, the heart. Sorry, this is right. This is left. Okay, and this is why this part is called what do you call it? Right atrium. Sorry, what do you call this one? Left atrium. They are the upper chambers. They are called atria. All right, this is right ventricle. Okay, lower chamber, left ventricle. Okay, okay. So blood enters the blood and then is pumped by the right atrium into the left ventricle. The left ventricle pumps the blood right into the, see the blood moving. Okay, let me use a better tool. Okay, watch this. So yeah, so the oxygenated blood, this is vena cava. The oxygenated blood into the right atrium pumps the blood. Now two of them are doing the same at the same point, at the same time. Oxygenated blood is entering here through the pulmonary vein. So when it comes here, so both of them contract at the same time, and then when the atria contracts, called atrial systole, the ventricles are relaxing to receive the blood. Because they are relaxing to receive the blood, the pressure here and here in the both atria are greater than the pressure in the ventricles. So the valve open, okay, so that blood can flow from, of course, from here, left atrium to left ventricle, right atrium to right ventricle. Okay, now, but you see, the question is asking us, what is the state of valves in the mammalian heart when the pressure in the ventricle reaches its max? So when the ventricle starts contracting, okay, these two valves will close. These are the atriovascular valve. This is a bicuspid valve. All right, both of them are called atriovascular valves because they prevent backflow of blood from the ventricles to the atria. So they will close so that blood does not flow back to the atria. Is that okay? Now, these two valves, these seminal valves, all right, aortic, this is pulmonary seminal valve, this is aortic, they will open so that blood can move, blood from the left ventricle can enter the aorta, this aorta, okay, and blood from the right ventricle can be pumped into the pulmonary artery, okay. So when it contracts, seminal valve will open, okay, and when the valves will close, this will close. So open and close, open and close. Seminal valve opens, okay, they will open, this will close to prevent backflow. So our answer is C. Normal venous pressure in the feet is in 3.3 kilopascal. When a person stands very still, venous blood pressure in the feet rises. When it stands still, okay, what causes high pressure? Okay, now pause this video and try it out. So A says muscles in the wall of the veins contract, reducing diameter of the vein. Now, the muscles on the wall of the vein, they are smooth muscles. They do not contract voluntarily. They are involuntarily, all right, stimulated to contract. So when someone stands up, standing up voluntarily. So of course they don't contract because the person is standing up. Is the brain that controls that. Okay, if they have to contract. So A is out. B says skeletal muscles in the legs. Okay, this is squeezing blood, not remove this, not this typo. Squeezing blood upward. Okay, in the okay, all right, because we're talking about normal vein pressure in the feet is this: when a person stands very still, okay, of course, pressure in the feet rises to 50 kilopascal. What causes the high pressure? Now this is pressure in the feet. Remember that if somebody stands still, okay, yeah, I think this is okay. If somebody stands still, okay, what happens is that the blood settles on their feet. Okay, now if this is contracted, if this, why is blood settling on their feet? Yeah, this is correct. Okay, if this is contracted, right, what's going to happen is that the blood will move upward towards the heart. But if it is not contracting, the skeletal muscles in the legs are not squeezing blood, so they are not squeezing blood upward in the veins. Okay, so if they're squeezing blood upward in the veins, they will not settle on the feet. Okay, so they're not working. But if they are working, blood will go up, right? So, but this says systolic blood pressure increases. Okay, standing up does not increase systolic blood pressure. Systolic blood pressure is actually affected by the left ventricle when it contracts. Okay, it has nothing to do with the feet. Seminal valves in the veins of the legs cease to function. They don't cease to function. Okay, they're not because they prevent backflow of blood. Okay, they maintain one-way flow. So it's just that when you stand up, okay, it's not. But once you start moving, the muscle starts contracting. We are standing still. There's a skeletal muscle in the legs are not okay. Why? Because look at what I said here. When a person stands very still, it's not moving. It's when you start moving, your skeletal muscles start contracting, and then blood will move up, right, towards the heart, because the skeletal muscles will squeeze the blood up. But if you're standing still, skeletal muscles are not contracting. It's moving that causes contract. All right. Therefore, the pressure in the legs will build up. Okay. So B is the most likely answer. Okay, 10 says which feature enables the aorta to withstand high pressure? Please answer at ventricular system. Please answer this question. I've answered it before. Right? There are two features that prevent it from busting. Right? Two features. Aorta is an artery, it's the largest artery for that matter. Right? Arteries have thick walls. The outer one has collagen and elastic fiber. The middle one has collagen, elastic fiber, and smooth muscle. So, but it's collagen and elastic, fibers that prevent it from busting. So, collagen, fibers, elastic fibers, right? These two prevent it from busting. Don't forget that collagen, all right, is a structural protein. Okay, it has high tensile strength that resists withstand large pulling forces. So, is A. Okay, so this is the end of the class for today. So thank you so much. All right, please ask your questions, subscribe to the video, follow us, all right, and make sure you share the video. All right, this is where we stop today. Thank you. Bye.